Hello everyone, my name is Brandon Lukas and I am a third-year PhD student in the Bioinformatics Department at the University of Illinois at Chicago. I conduct my research under the advisement of Dr. Yang Dai, focusing on understanding gene regulatory networks. Today I will be presenting my research on integrating ENCODE and context-specific H3K27ac ChIP-Seq and RNA-Seq data to infer transcription factor activity. As we know, transcription factors are crucial regulators of gene expression and play essential roles in cellular and disease biology. Dysregulation of transcription factor activity can lead to various diseases, such as cancer and uterine fibroids. However, current methods for predicting transcription factor activity face limitations, including the reliance on curated gene regulatory networks and failure to integrate multi-omics data. Our main objective is to develop a novel, network-based method for predicting context-specific transcription factor activity by integrating transcriptome and epigenomic data. Our auxiliary objective is to assess the performance of this method in accurately predicting context-specific transcription factor activity and identifying novel, biologically relevant transcription factors. We used a uterine fibroids dataset as a case study for developing our tool, but it is designed for broad applicability across various biological conditions. We worked with RNA-Seq and H3K27ac ChIP-Seq data from leiomyoma and matched adjacent myometrium samples taken from 20 patients, and used the ENCODE database in our analysis. To infer transcription factor from gene expression data, we employed network-based statistical methods, including a U-cell, GSEA, univariate linear model, and weighted sum model, with either the full transcriptome or a subset of differentially expressed genes as input. In our proposed integrated framework, we combine H3K27ac ChIP-Seq data with the ENCODE database to create a context-specific network called the Context ENCODE Network. The construction of this network involves the following steps. First, we utilize the H3K27ac ChIP-Seq data as a filter for the ENCODE database. H3K27ac is a histomodification that marks active regulatory regions, and its presence can provide valuable information about the regulatory landscape of the specific biological context. Next, we map the ENCODE transcription factor binding locations onto the H3K27ac marked promoters. This allows us to identify transcription factors that are more likely to be active in the context of interest, as they are bound to active regulatory regions. Once we have mapped the transcription factors to their respective promoters, we associate them with their nearest target genes. This process helps to establish regulatory interactions between transcription factors and their target genes, giving us a context-specific gene regulatory network. This network is then used with either the full transcriptome or differentially expressed gene subset RNA-Seq data to infer transcription factor activity using network-based statistical methods such as a U-cell, GSEA, univariate linear model, and weighted sum model. By leveraging both transcriptome and epigenomic data, our proposed integrated framework can provide a more comprehensive and accurate understanding of transcription factor activity in various biological processes and disease states. Comparing the Context ENCODE network with Dorothea, a widely used curated gene regulatory network, we found similarities in the number of transcription factors and target genes. However, the Context ENCODE network has significantly more network connections and a higher average number of targets per transcription factor, indicating a more dense network. We also compared tissue separation at the differential gene level using Dorothea and Context ENCODE networks through hierarchically clustered heat maps based on the top 30 differentially active transcription factors. The heat maps showed differences in the top 30 differentially active transcription factors, suggesting the potential influence of the choice of network on transcription factor activity predictions. Although the clustering patterns in both are very similar and both are capable of separating leiomyoma and adjacent myometrium tissues. We also present inferred transcription factor activities of key transcription factors implicated in leiomyoma biology for 4 methods, utilizing Dorothea or context ENCODE network at either the full transcriptome or differential gene level. The accompanying table summarizes the specific roles and implications of each transcription factor in leiomyoma biology according to the literature. You will notice there isn't much consistency in these results, and some predictions are missing. Our context-specific network-based approach integrates large-scale databases and context-specific multi-omics data. And while current subjective evaluation methods make it difficult to determine the enhanced transcription factor activity predictions, we currently plan to develop a quantitative evaluation procedure for a more objective assessment. Nonetheless, our integrated framework can help generate testable hypotheses, potentially improving the understanding of the molecular mechanisms behind certain diseases and help identify therapeutic targets for treatment. This project was supported by a T32 grant from the National Cancer Institute, and I want to thank you for your attention, and I would be happy to answer any questions you may have.